Hello friends and viewers. Today in this video I am going to talk about how coronavirus or the SARS-CoV-2 virus infect the human lung cell and how this virus replicate inside of the lung cell. Due to the pandemic nature of this virus, the virus is spreading all over the world. So it is very good to know that how this virus grows inside of our lung cell. First of all, let me show that how this virus transmit from one person to another person. They transmitted through the cough, sneeze, touch, and the fecal oral contamination. And then they travel to one person to another person through the eyes, nose, and mouth. And then they can infect the oral epithelial cell, ciliated cell, and type 2 secretory cells. And when they infect the cell, then there are several steps the virus take places so they can multiply inside of the lung cell. For example, just imagine this is the lung cell and this is the virus. So step 1 is the attachment of the virus. So the virus attaches to the human lung cell specific protein that is called angiotensin-converting enzyme 2. So this angiotensin-converting enzyme is the receptor of this virus. But the question is that why this virus actually bind in this receptor? Because they have the structural homology. So the angiotensin converting enzyme 2 and the viral spike protein has the structural similarity. That's why they bind to this receptor. This angiotensin converting enzyme 2 is highly expressed by the oral cavity epithelial cells and the secretory cells like the type 2 pneumocyte inside of the lung. And when this virus bind through the ACE2 by its spike protein, another enzyme that is expressed on the human lung epithelial cells, transmembrane serine protease 2, which cut a part of the spike protein, which facilitate to entry of this virus inside of the lung cell. Not only this serine protease is involved in this process, but many other proteins has been described that can help entry of this virus inside of the lung cell. The process of this virus enter inside the cell is called endocytosis. The second step is the uncoating of positive sense single-stranded RNA. Since you know that the virus is composed of the spike proteins and some other structural protein, but it has the single-stranded RNA as a genome. So in this step, the virus uncoat this single-stranded RNA. So now the single-stranded RNA in the cytosol of the human lung cell. And this single-stranded RNA mainly comprises of 30,000 base pair compared to our own genome is 3 billion base pair. And this virus's mRNA can produce at least 29 protein up to date. And this mRNA has multiple open reading frame, especially the open reading frame 1A and the 1B that can produce the polyprotein in the next step. And it has also the structural protein like the spike protein gene, envelope protein gene, membrane protein gene, and nucleocapsid protein gene. These 4 genes product or the protein is called the structural protein, and rest of the genes products are called the non-structural protein. They are mainly help to grow inside of the cell, but they do not go inside of the virus after the replication. So the third step is the polyprotein translation. That means that from these RNA, our cells, ribosome, translate a polyprotein. Polyproteins means that multiple protein attached together. Since this virus RNA is single-stranded positive sense RNA, so our ribosome can treat it as a messenger RNA. Since you know that only the messenger RNA can be translated through the ribosome, so our cell's ribosome cannot distinguish from the viral RNA from our self RNA. So they actually produce the polyprotein. This polyprotein has multiple protein attached together, and one of this protein is RNA dependent RNA polymerase, and some accessory protein, those help for the replication of this RNA. So this RNA dependent RNA polymerase and the helping protein are called the replicase. So next come the fourth step, the RNA replication. So this RNA dependent RNA polymerase and the helping protein help to synthesize the next multiple RNA. For example, the RNA for spike protein, envelope protein, membrane protein, and nucleocapsid protein, and other non-structural protein. And it can also replicate from its 3 to 5 prime strand to 5 prime to 3 prime positive sense RNA. So all of these RNA then translated to its proteins, which is the fifth step of this virus production. So these proteins are mainly translated in the rough endoplasmic reticulum. Normally, the endoplasmic reticulum contains the ribosome that is seen here. That's why they are called rough endoplasmic reticulum. So then these proteins are assembled into a virus particle with its RNA and nucleocapsid protein. These structural proteins are transported by the Golgi apparatus, and especially the spike protein help to assemble the structural protein with its single-stranded RNA. So that's how they finish the sixth step, or the assembly of proteins and RNA. Then they release from the cell by budding process, or just a reverse process of the endocytosis, that how they enter inside the cell, called exocytosis. This is how the SARS-CoV-2 virus replicate inside of the human lung cell, and after that they can also infect the nearby cell or any other cell, those express the angiotensin-converting enzyme 2. Thank you for watching. Hope you have understand that how that coronavirus infect our lung cell and replicate inside of our lung cells.